Marcella Birtele also from USC, talking about non-synaptic function of ASD-associated gene SYNGAP1. Thank you.
Hello, everyone. So I wanted to start my presentation showing you what we mainly use in the lab. So what you see here is a brain organoid. And we use this model system to dissect and better understand neurodevelopmental disorder. So in Giorgio Quadrato's lab at USC, we really want to try to look at the molecular mechanism behind the neurodevelopmental disorders. And as we know, NDDs have a very heterogeneous clinical phenotype, also have a very heterogeneous neurobiological neurobiology, and also a huge diversity for their genetic causes. So if we look at NDDs, we know that most of these disorders are polygenic disorders. And this feature is really difficult to model using rodents or animal models. And that's why we mainly use human-derived stem cells to dissect the genetic components of these disorders. And also we can look at the genetic background and the effect of the genetic background on different variants. So really what we do in the lab is to follow and take inspiration from what happens in vivo. So as you see here from the left side, we have that in vivo after gastrulation, there is the generation of 3 different germ layers, where the ectoderm will generate the neural tube that upon folding will generate different parts of the nervous system. And with a different concentration of morphogens, this neural tube will develop and specify in different areas. So what we do in the lab is to take mainly iPSC cells, and differently from 2D cultures, we leave these cells in low attachment mode, in the sense that these cells will self-aggregate and self-organize. So we leave the cells to really mimic the site architecture of the developing cortex. And what we do then is to apply a different concentration of gradients, here TGF-beta, BMP or Wnt morphogens, to really push the differentiation towards a cortical fate. So the specific protocol we use in the lab is an adapted protocol from the Arlotta lab, where we can generate all the main cell types that we have in the developing fetal cortex. So, and this is really important in the context of modeling NDDs, especially because in the human cortex we have specific cell types, such as the outer radial glia, that are not present in rodent models or other animal models. And here you also see a single cell sequencing analysis from different batches of differentiation for these organoids. And you can see that there is a highly homogeneous generation of the different cell types. So with this model, when I joined the Quadrato lab, being an electrophysiologist, what I wanted to do was to look at the function of SYNGAP in excitatory neurons in cortical organoids. As we know that mostly all the work that has been done so far has been described SYNGAP in mature excitatory neurons in rodent models and only recently in iPSC's 2D models. So we started to look at the SYNGAP expression in some fetal data set. So as you can see here from the left side of the slide, we have that if we look at the SYNGAP1 RNA expression over different post-conceptional days, we have an increase in expression of SYNGAP, and this is in line with the already known function of SYNGAP in mature neurons. But what was really surprising to us was when looking at the Syngap expression in the different cell types, we could, of course, see the Syngap expression in the neurons, but strikingly was the expression of Syngap in radial glia cells. So this was completely really surprising for us, and we wanted to confirm these results at the protein level. So we went to the organoid system and we waited 2 months. We cultured these organoids, and at this time point, what you can see is a SOX2 positive area in the left side, upper left side of the panel, where SOX2 labels radial glia cells. So this rounded structure are really mimicking the neural tube that we previously saw in the fetal embryo. And so we wanted to look at the Syngap expression, and within these radial glia cells, here in red, we can see Syngap expression in the apical domain of the Syngap, of the radial glia cells. So we asked also the COBA lab for some help with the proteomic, and we used day 7 organoids this time for having a homogeneous population of radial glia cells. And we confirmed the presence of Syngap peptides. And we also saw that among the interactors of Syngap, we could find TJP1, which is a tight junction protein that you can also see here in white in the stainings. That is really important for the maintenance of the ventricular wall. We also look at the isoform present in the radial glia cells, and we found that alpha-1 Syngap isoform was present in the radial glia cells. We then wanted to look at the function of Syngap in these radial glia cells. And from here, I completely forgot about my first project as I joined the lab, and we really focused only on radial glia cells. So we compared day 7 organoids from a control line with day 7 organoids from a cell line carrying a truncating mutation, giving rise to haploinsufficiency for Syngap. And what we saw from bulk RNA sequencing was that mainly the differentially expressed terms were related to cytoskeletal function. And also, when looking at the interactors of Syngap, most of the proteins that are interacting with Syngap in radial glia cells are related to cytoskeletal remodeling. And if we think about cytoskeletal remodeling in radial glia during the developing cortex, we know that this is a really important feature of this type of cells. So here you can see the developing cortex with the radial glia in blue, which extend 2 different cytoskeletal processes. One apical process that goes and really help tightening the ventricular wall of the ventricles. And in this area, the radial glia cells will proliferate. And once the radial glia proliferate and generate neurons, these neurons will then migrate onto the other domain of the radial glial cells that extend toward the basal side. So, and this will allow for the proper positioning of the neurons and proper lamination of the cortex. So we wanted to look at this phenotype and function of Syngap from the cytoskeletal point of view. And we use a 2.5D system where we're able to recreate this ventricular generation. So here what you see is that the cells are organizing around the ventricle, also called rosette-like structure. And what we see here is that the patient line generates a much larger and disorganized rosette. And upon quantification, we could see that the patient-derived line generated fewer number of rosettes. And at the same time, the generated rosettes were more irregular and also with a larger ventricular wall. So going back to my introduction, we also wanted to look at the effect of the background, of the genetic background. So we used a line generated from the COBA lab where the same mutation was inserted in a control background. And what we could see was that the extent of the impairment in the rosette was similar to the patient line, indicating that the background in this case does not have an effect on the SYNGAP function at the cytoskeletal level. We then also used another line carrying a mutation that gives rise to a non-functional domain for the RASGAP called RASGAP dead. So in this case, here in the Q panel, what you can see is that the phenotype was even more exacerbated. So almost no rosettes were generated from this line, indicating that the RASGAP domain is essential for this cytoskeletal function in human radial glia. So then we looked, as I said, radial glia cells have also this function of guiding neurons and generating a laminated cortex. So here you can see a 2-month-old organoid from the control line, where we can see a clear separation from these blue cells that are the radial glia cells compared to the neuronal population that is labeled here in green and red. So there is a proper lamination process happening within 2 months of cultures. But if we go in the SYNGAP patient line, we can see that this organization is completely disrupted, suggesting that human radial glia have this impairment in guiding neurons to the proper position. So we also wanted to check at radial glia in this organoid context. And we looked at their division mode. As I said, radial glia, when they're close to the ventricle, they're highly proliferative. And they can self-renew or also generate neurons. So what we saw was that in the SYNGAP patient line, we had an increase in differentiative divisions, suggesting that there is an imbalance between progenitor and neuronal populations. So we used single-cell sequencing analysis on 2-month-old organoids from control and patient-derived lines. And we really zoomed in the apical radial glia cell population here in yellow. And what we saw was that if we look at the terms that were expressed in apical radial glia in the SYNGAP patient line, we had a complete downregulation of self-renewing genes, and there was a clear upregulation of neuronal genes, really suggesting that there is an effect on the division mode of these radial glia cells. So we performed a quantification of the total number of SOX2 positive cells, so progenitors and neurons. And what we could see also in the other lines was that the patient-derived lines had a decrease in the progenitor population and an increase in neurons. And so upon these quite striking results, we had actually a lot of pushback from the reviewers and also upon grant submissions. So we were asked to add on this human data, also some mouse analysis. So we looked at E18.5 mouse sections from het and knockout of a SYNGAP mouse model. And what we saw here was also a striking decrease in the SOX2 positive area, so in the progenitors, coupled with an increase of the neurons. And this also, going back to the previous publication, we also know from the work from Helen that this was seen also in Xenopus. Suggesting that this phenotype and this role of SYNGAP in progenitors is maintained across species. And finally, we also looked at the neuronal phenotype. And we could see from single cell sequencing that I'm not showing here at 4 months, and calcium imaging as well as arborization and structural analysis that in Syngap patient-derived lines, we had an accelerated maturation in line with previous results in rodents and human. So just to conclude, we showed a novel function of Syngap in human radial glia progenitors. And with haploinsufficiency for Syngap, we see a disruption in the cortical plate organization as well as in the ventricular maintenance. And this is coupled also with an accelerated neuronal maturation. And finally, just a few words. What we are doing now is to try stratifying on these results. So we're increasing the number of variants and the patient-derived lines. And we're generating rosettes, and really we want to try to dissect the effect of different variants and also the effect of different variants on different domains of the protein. And with this, I want to thank especially Dr. Quadrato, who was essential for guiding this project. Also, Ashley Del Dosso, who's a co-author on the study, all the lab members, all the collaborators, and all the funding agency. Thank you.